So yeah, I'm happy with you recording it. You're recording it now?
Now we're recording, yep.
Okay, well, thank you for inviting me to give this talk. I understand Dr. Barb, my colleague for a long time on this project, has given you a talk recently, and she's been invited back to do another one.
And she didn't cover this subject area. We just did basics of well-area degeneration and axon regeneration.
Okay, so I'm not going to repeat what she said. That's good.
No, you're safe.
Plus, I don't know as much about it as she does. So this is a project that I usually show this slide first with all the collaborators, and it doesn't include everybody, but this kind of a project requires an army, and this is just a partial list of the people. I have bolded my longtime colleague, Dr. Mary Barber, you've met. And this character here, Justin Brown, has been very instrumental in getting this project moving along for long clinical translation. And I'm really much indebted to Justin for this. So let me just go over some of the milestones over the last, we've been on this for about 20 years now. It started in about 2000 with Dr. Randy Betts as the chief of Shriners Hospital here in Philadelphia, who had the idea to reinnervate the bladder using an intercostal nerve to move it down to the pelvic plexus and reinnervate the bladder. And we got funding from the Shriners Hospitals to do that. Turns out that was not feasible. The first surgery we did, the dog nearly died. We had to paste two intercostal nerves together, and it actually worked on that one side. The dog almost died. And so we quit while we were ahead. And when we were doing that surgery, an orthopedic surgeon working with us from the Shriners, Linda DeAndrea, well, we have another nerve right next to us here that innervates the tail. He doesn't need to wag his tail. Let's use that one. So on the other side, we did a nerve transfer with the caudal coccygeal nerve to the bladder nerve, the S2 root in this cord, in the sacral cord. And so we did a whole other series of experiments using that, and so we changed the title to somatic nerve transfer using the coccygeal nerve. And then, as Justin said, I was invited to give a talk by Ken Peters at the Beaumont Hospital in Royal Michigan, on where he was collaborating with this Chinese urologist. And that trial actually that Ken Peters did was stopped because of some issues that Xiao got involved in that I can't get involved in now, but I could talk to you about it later. But that's when I met Justin Brown, and I was very interested in working with him. A lot of people came up to me and talked to me about it, but no one actually ever followed up. Justin's the only guy that ever followed up, so it's just, I really owe him a lot to see this through to clinical translation this month. Mary Barb had been collaborating with us during that time, but she wasn't funded for it, so she got kind of tired of that. When we put into a multiple principal investigator application to the National Institute of Health, the National Institute of Neurological Disease and Stroke, and that's been funded for 3 consecutive 5-year funding cycles, and culminating this month in the first nerve transfer that's scheduled up at your place. So let me just first kind of refamiliarize you with the innervation of the bladder that you may have forgot about since medical school. The bladder is a parasympathetically dominated organ, or autonomic system dominated organ that receives parasympathetic innervation from the sacral cord through the major, in the rat it's called the major pelvic ganglion, but because all the cell bodies in a rat exist in this ganglion here, but in the human and all other species it's called the major pelvic plexus. And then fibers emanate from that to innervate both the bladder and the bladder neck and the bladder dome, and some innervate the smooth muscle of the urethra. And then there's the pudendal nerve, that's a somatic nerve that innervates the external urethral striated sphincter as well as the anal sphincter. And then there's the sympathetic nerves that innervate the ureter as well as the bladder dome. And the bladder dome, they release norepinephrine that primarily acts on beta-3 receptors to mediate bladder filling. whereas the parasympathetic nerve it releases acetylcholine to act on both M2 and M3 receptors, but primarily M3 receptors mediate the contraction. The M2 is sort of a backup contractile receptor that we've been working on for 15, 20 years. Whereas the norepinephrine released in the bladder neck, because of the higher density of alpha-1 receptors, maintains contraction of the bladder neck, and during filling, that keeps the bladder neck closed and allows filling of the ureter of the bladder. And of course, the somatic nerve, it releases acetylcholine acting on nicotinic receptors on the striated muscle of the external urethral sphincter. So I want to make a little bit of a distinction between upper and lower motor neuron lesion. And when these were originally terms that were used for somatic nerves, where you have a upper motor neuron coming out of the motor cortex of the cerebral cortex, going down to the ventral horn of the spinal cord, synapsing with a lower motor neuron that actually leaves the spinal cord and innervates the skeletal muscle. So there's a two neuron system in this sympathetic and parasympathetic autonomic nervous system. It's more than just two neurons. So it's a little bit of a misnomer with the bladder, calling it an upper motor neuron lesion. But anatomically, if you have a spinal cord injury above the sacral cord, you have a different clinical outcome. You have hyperreflexia, where the bladder contracts during filling, and you get what urologists call detrusor sphincter dysynergia. That's defined by urodynamics, so the bladder contracts during filling. And the striated sphincter also contracts as the bladder tries to empty. And this causes a lot of problems with high pressure voiding that can lead to renal damage. But we're not dealing with this phenomenon. We're dealing with a lower motor neuron lesion where it's below the L1 or T12. It affects these spinal cord segments, the sacral 2 to 3, 2 to 4, primarily 2 and 3 in humans, that actually innervate the bladder, the motor neurons that innervate the bladder, as well as conus medullaris and caudate equina lesions. And it results in a flaccid bladder paralysis because there's no connection between the bladder and the spinal cord or the central nervous system. And this happens with lap belt injuries. And about 25% of the patients at Shriners Hospital have these lower motor neuron lesions. And these patients have no treatment right now. They require, if you want to make the bladder work with the brain, it has to have new nerves to reinnervate. And that's what this whole project has been about. Now, we use the dog model, and the dog has 2 extra lumbar vertebrae. Humans only have 5 lumbar vertebrae. That allows them to twist around, and it's kind of a hassle for us because they can actually chew on their tail when we cut the tail nerves. So we always we amputate the tail after we do these surgeries. So the original model was a decentralization of the bladder by doing a laminectomy at L7, a complete laminectomy at L7, a partial laminectomy of L6 and the S1, where we can identify the roots coming out of the cord. Now, this is a diagram from the Miller's Anatomy of the Dog book, and they have the dura opened up here. But we never entered the dura intentionally when we do these surgeries, so all this is done extradurally. And we can stimulate these nerves and measure bladder pressure and identify the nerves that cause the bladder to contract, and then sever them. And that's our model of decentralization. So the first set of experiments we did were to identify the roots on each side that induce bladder pressure with electrical stimulation. Now, this diagram, we never did this on any dog. On this left side, these are diagramming the coccygeal nerve transfer, where we took a nerve that normally innervates the tail and transferred it to the S1 or S2 root that innervates, that causes the major contraction of the bladder. And in those experiments, we actually put an electrode around the donor nerve, and we had nothing but problems with electrodes, but I'm not going to talk too much about that today. And in another group of animals, we did the nerve transfer intra-abdominally. We took the genitofemoral nerve that its anatomical path before it leaves the pelvis runs right close to the pelvic plexus. And so we were able to transfer that nerve to the major nerve coming out of the pelvic plexus, going to the bladder, which actually doesn't have an anatomical name, an anatomical textbook. So we name that nerve the anterior vesicle branch of the pelvic nerve to identify it as the nerve that's coming out of the pelvic plexus that goes to the bladder dome and mediates contraction. And we did these transfers either immediately after the decentralization in the spinal cord, or at 1 or 3 months after nerve transection, this sort of surgery. And then we did the assay of whether the bladder could contract in response to these nerves with retrograde tracing, as well as stimulation intraoperatively right before euthanasia, 1 year after the nerve transfer. And these are intraoperative photographs of this. And I should say that we consulted with pediatric urologists who advise us that the way they manage these kind of patients is they create a abdominal vesicostomy. They open up the bladder to the abdomen and allow the bladder to empty into the diaper. And so we did that on these original dogs. Turns out that wasn't necessarily required. So let me orient you to this photograph. This is the top of to the right is the cranium, to the left is the caudal. And here is the anterior vesicle branch of the pelvic nerve coming out of the pelvic plexus going towards the bladder. And here is the genitofemoral nerve coursing down, and we took the entire genitofemoral nerve. It doesn't have much of a function that's that useful. It's involved in the cremasteric reflex. And so it's a nerve that can be sacrificed without much functional deficit. And this is where the actual apposition of the genitofemoral nerve to the anterior vesicle branch of the pelvic nerve coming out of the pelvic plexus and going to the bladder. And what we do right before euthanasia, here's the Foley catheter going into the bladder dome through the vesicostomy. We stimulated with this Medtronic's intraoperative stimulator. You may recognize this. And that, if we stimulate the anterior vesicle branch of the pelvic nerve, it causes the bladder to contract. And we can stimulate it before or after the anastomosis of the donor nerve. And so these initial studies, we compared the genitofemoral nerve, which is primarily a sensory nerve. Actually, there aren't any nerves in the body that are 100% sensory or 100% motor. They're all mixed. But the genitofemoral nerve is primarily sensory. And we compared that with branches of the femoral nerve, which is primarily motor. And it turns out if we use the genitofemoral nerve, which is primarily sensory, we get less return of, less contraction of the bladder by stimulating that transferred nerve than if we use the primarily motor nerve, the femoral nerve. And so that makes sense. There's more motor axons in the femoral nerve than there is in the genitofemoral nerve, and it primarily is 1-alpha big motor neurons that normally innervate the skeletal muscle of the leg.
And Mike, a quick interruption. Did you talk at all about the hybrid synapse? The fact that this is somatic cholinergics that are now interacting with the muscarinic parasympathetic cholinergics.
Yeah, so normally the femoral nerve innervates the skeletal muscle, and it releases. They both release acetylcholine, but the receptors are different. The receptors are nicotinic in the skeletal muscle, and they're muscarinic in the smooth muscle. And we think that these new nerves, as they grow in, they kind of change their phenotype a little bit, and they're still releasing acetylcholine. But now that acetylcholine is now acting on whatever it innervates. And it innervates, we've done studies to show that the new pathway innervates both the smooth muscle directly, which normally is not innervated, but it also innervates the intramural ganglion cells, which are the pre-neuromuscular junction neurons that are in the bladder wall in most species other than rodents. And I think what happens for long term is that those are the active neurons that mediate the contraction by the new pathway. because direct innervation of smooth muscle by the new pathway is maybe not as functional as the reinnervating the intramural ganglion. But that's just a supposition at this point. We have no evidence to support that. So in one of the first animals that we did this vesicostomy and did the nerve transfer, and then what we decided to do is to reverse the vesicostomy to see whether the animal can empty its bladder on its own. And what we found that the vet tech that was taking care of the animal after we did that, this is right immediately post-operative to the vesicostomy reversal. So the bladder can only empty through the urethra now. It was normally emptying through a opening in the abdomen. Let's see if I can get this to play. This is a movie. This is what the dog did after we reversed the vesicostomy, immediately post-operatively. So this is was very reassuring. We can see the dog actually emptying his urine coming out over here. So this was very exciting to us. Looks like we solved the problem. Got this dog that has been decentralized and now it can empty its bladder. So these initial studies, we showed that we can reinnervate the bladder by either cutting the bladder nerves and immediately repairing the same nerve that innervates the bladder, or transferring a primarily somatic nerve, the coccygeal nerve, to the severed bladder nerves that was actually in the sacral cord where we did that nerve transfer, or transferring a genitofemoral or femoral nerve to the anterior vesicle branch of the pelvic nerve in the abdomen, either immediately 1 month or 3 months after bladder denervation. And well, it turns out that the primarily somatic motor nerve, the femoral nerve, seems to provide a little better return of nerve-evoked detrusor contraction than the primarily sensory nerve, the genitofemoral nerve. And Mary Barb is a really highly skilled neuroanatomist, and we've done these retrograde tracing studies with her, where you can inject the bladder 3 weeks before euthanasia with these fluoro gold, and there's a couple of other different colored retrograde dyes. It's picked up by the nerves and they're transported retrogradely to the cell bodies wherever these nerves are. And so this shows the femoral nerve transfer where the bladder is here, and here's an anterior vesical branch of the pelvic nerve. This actually shows the fluoro gold in the nerve in a gross photograph. So the fluoro gold is being retrogradely transported across the anastomosis site back to the cell bodies. Here it shows proximal to the suture, the actual suture of the like the 9-0 nylon suture were used to actually attach the neural lemma of the two nerves. It's shown right here. And so you can see this fluoro gold very clearly showing neuroanatomically that there's reinnervation. And we trace these nerves all the way back to the spinal cord. Now this is a normal innervation of the bladder over here from if you inject a normal dog or a sham operated dog. This is the ventral horn of the sacral cord. In this lamina 7 is primarily where the bladder neurons, the motor neurons of the bladder exist. After the genitofemoral or femoral nerve transfer, the retrogradely labeling from the bladder labels motor neurons in the spinal cord at a different lumbar left segment, the lumbar, L3 to L4, as opposed to S2 and S3 in the normal dog, as well as in different lamina of the spinal cord, lamina 8, 9, and 9 here. So this kind of proves neuroanatomically that we have reinnervated the bladder with the donor nerve, because it comes in, the retrograde dyes label the spinal cord at a different spinal level and a different spinal lamina, typical of the genitofemoral or femoral nerve. And what Mary has done is actually count the number of cells per cross-sectional area of the section of the spinal cord at all these different levels. And so this is the normal innervation of the bladder, the motor innervation bladder, probably S1, S2, and S3. There is some lumbar direct innervation of the bladder. This is not well accepted in the literature, but we've done this a number of times, and this is clearly there's a lumbar direct innervation of the bladder. When we cut the everything below L7, the S1, 2, and 3 roots, we've eliminated this. With the genitofemoral nerve transfer, this is the origin of the genitofemoral nerve, and so that shows that these pathways have been reestablished with the nerve transfer. And this is femoral. This is the femoral nerve transfer. So what about sensation? Well, this is the motor control of the bladder, but can the animal tell that the bladder is full? What we did initially was retrograde tracing of this dye to the sensory innervation of the bladder, the dorsal root ganglia. And so Mary's lab actually counted the number of cells per cross-sectional section area at each of these dorsal root ganglion spinal levels. And we have lots of data here, so it's a little busy slide. This is showing what they look like, what the retrogradely labeled dorsal root ganglion neurons look like in the, here's the sham sacral ganglion. And when you transect the sacral root, you don't see this labeling. And here's the genitofemoral nerve transfer up in the L2, dorsal root ganglion. So normally in the open parts here, these are the normal innervation of the dog bladder, L6, L7, S1, and S2, and a little bit of S3. When we do the nerve transfer, these are pretty much gone, as well as in gray. This is the control, just transected. So this is exactly what you would predict if you did a complete transection of the dorsal and ventral roots of everything below L7. And then we started putting a camera over the dog's housing cages and video recording their behavior over 24 hours and counting the number of times. And these are all female dogs, so they're much easier to catheterize. And so they show this characteristic squatting behavior. If anybody's walked your dog, you know he's going to do number 1 or number 2 based on their posture. And so we counted those number of postures per day, and this is the normal number of postures, number of micturition or defecation postures. And then after genitofemoral nerve transfer, we actually had an increased number, or the femoral nerve transfer, we had an increased number. So this evidence could be interpreted that the reinnervated animals can detect bladder fullness through this new neuronal pathway we created by the nerve transfer, except that if the decentralized only animals also display these behaviors, that would be less secure a conclusion, because we hadn't done any of these videoing of the decentralized only. Well, then we did that, and it turns out they do. So if we cut everything where the L6 and L7 dorsal roots are intact, and this is a diagram of the spinal column that we cut everything below L7. And in these dogs, we also cut the hypogastric nerve, providing sympathetic innervation. So we cut the sympathetic innervation to the bladder. And this is an individual graph for each dog that we did. On the x-axis is the number of days after we did the decentralization, and in blue is the number of voiding postures per day, and red is the number of defecation postures per day. And you can see in all 6 of these dogs, it had no effect, or maybe even increased the number of voiding postures. So what we concluded from this is that the L6 and L7 dorsal root possibly participates in bladder fullness sensation. And so this is what we're talking about here in these in the gray and the sham operated. L6 and L7 has a number of neurons that pick up the dye from the bladder and transport it back to the dorsal root ganglion, as well as in the genitofemoral or femoral nerve transfer animals. Whereas we don't know whether in the nerve transfer, these neurons up above are mediating the bladder fullness sensation, or is this the remaining fullness sensation detected by this L6 and L7. So we did a whole nother series of 8 animals, along with an army of medical students one summer, where we cut both the L6 and L7 dorsal roots, as well as the hypogastric nerves and everything below L7. And these are those similar graphs as I showed in the previous slide of the number of postures per day. Now she's also Ekta Tawari, the graduate student at the time, with doing these video monitoring. This is a really busy slide, but it turns out that when we cut the sensory roots, these animals didn't do well, and we couldn't keep them alive. The veterinarians demanded that we euthanize these animals early, and 2 of them we had to do it within 2 months. But every dog that we did this to had a large number of positive urine cultures, and most of those urine, pretty much all of those urine cultures were associated with increased numbers of white or red blood cells in the urine, or gross hematuria. None of them had a fever, but since they don't have sensation, definition of a urinary tract infection really is based on symptoms, and they can't display symptoms if they don't have sensation. So we're stuck with using laboratory values of the white blood cells and red blood cells in the urine as a measure of whether they have a urinary tract infection. So all these animals really had urinary tract infections. And one of these animals, this is number 18, started showing behaviors that were kind of intermediate between urination and defecation. And this one we had to euthanize, even though it didn't show any urination postures. It had kidney stones, and we were ready to reinnervate this animal 1 year later. But we had to euthanize the animal because of all the stones that was developing. But this is the behavior that this animal shows that Ekta Tawari decided was intermediate between urination when the legs are splayed apart and it kind of squats, and defecation. So the animal's feeling something down there in the pelvis, but doesn't really know what it is or what to actually do about it, whether it's a defecation or a urination. So there is a new pathway that the animal's trying to interpret what to do about it. And so these are the, at euthanasia, about 6 months to a year after the nerve transfer, we do a laminectomy from the sacral all the way up to the T10 or even T9 sometimes, and we stimulate each spinal level, either the spinal root or the spinal cord, the entire spinal cord, and measure how much bladder pressure that produces. And you see in a normal dog, it's primarily these sacral root, but they do have a direct innervation of the L2. Now, some people would say this is maybe due to a hypogastric nerve, but we think this is a direct innervation, motor innervation at the L2, L1, L2, and L3 level in normal dogs. Kind of confuses it because this is the source of the genitofemoral and femoral nerve overlap a lot with these. So this is the dogs where we transected all roots called L7 and the hypogastric nerve. And so we didn't get any contraction down at the S2 and S3 roots. In one dog, we got a contraction that's simulating the S1 root, and it turns out that one we missed the decentralization. When we did the nerve transection, we couldn't find it. It was buried underneath, and we missed it. But normally, all these dogs that we did not show an S1 mediated bladder contraction. And you can see there seems to maybe be a little bit of sprouting of these nerves because there's higher contraction at stimulating these upper lumbar and lower thoracic levels. So here is the animals where we translated all roots caudal L7 and the L7 dorsal roots and the hypogastric nerve. And you can see that we got no contraction when we stimulated these spinal levels.
A quick question for you on this, Mike. The detrusor pressure is always going to reflect intra-abdominal pressure, and a lot of those are going to translate to the abdominal musculature. Do you think potentially you're measuring increased intra-abdominal pressure?
Yeah, that's a good question, and that's what a urodynamicist would ask. So I'm glad to hear you ask that. So we've always had a transducer in the actual rectum. So we're subtracting the vesicle pressure, which is a combination of abdominal pressure and detrusor pressure. We're subtracting the a reflection of abdominal pressure by rectal pressure. And this is how urodynamicists do it. We didn't have a pressure transducer in the actual peritoneum.
Okay.
But this is true detrusor pressure as urodynamicists define it.
Okay.
And then when we cut the L6 and L7 dorsal root, there's only 4 of these animals of the 8 animals we were able to study with this. I mean, these dogs were completely decentralized. They only had this minor direct innervation of the bladder by the L2 and L3 roots. These are the ones we wanted to study with reinnervation, but we just couldn't keep them alive long enough to do the nerve transfer. So from these initial studies, we found no evidence of urinary tract infection or stones in the animals that were decentralized by just cutting everything below L7, where the L6 and L7 dorsal roots were intact. In all the animals where we did the L7 dorsal root transection, just L7, we had culture confirmed bacteriuria that was coincident with alcohol blood or greater than 4 white or red blood cells per high powered field in the urine, indicative of a urinary tract infection. And in the animals that had both L6 and L7 dorsal root transections, 6 of those 8, we had infections, but the other 2, we only could keep alive 2 or 3 months, actually 1 or 2 months after. I suspect the other 2 would have been infected eventually too. So we had to conclude from this evidence that the bladder afferent innervation protects against bacterial colonization and maybe urinary tract infection, which is a whole different project that we hadn't even considered pursuing. And we're thinking about a way of pursuing that right now. But the afferent innervation of the bladder is important for protecting the bladder. Somehow, is it mucosal integrity or something about the way the bacteria adhere to the mucosa? It's a whole series of investigations that we're thinking about pursuing, but it's not really relevant to what we're funded to do right now. So the most recent experiments that we've done, and I only have a few minutes left, so I want to try to get through this. These are the, we have 2 papers that are coming out back to back, part 1, part 2 in the American Journal of Physiology, Integrative, Regulative, and Comparative Physiology. I just sent the galley proofs in, and this is the general design of that experiment. We have 14 controls, 11 sham operated and 3 normal unoperated controls. And then we have 4 animals that we decentralized. These are animals that we monitored their urination and defecation postures every month for the entire time, 18 to 21 months. And they never showed these postures after transection. All spinal roots, both dorsal and ventral, below L7, and the L7 sensory nerve and the hypogastric nerve. And it turns out that only about 60 to 70% of those animals don't show these behaviors. And then we had 8 animals where we did the same decentralization procedure and then did a nerve transfer using about 25% of the fascicles of the obturator nerve and transferred that to the anterior vesical branch of the pelvic nerve. These animals didn't lose any, some of them lost a little bit of ability to keep their hind legs together because of the loss of those fascicles, but that recovered. And so the obturator nerve still was functional in controlling the movement of the leg in these animals. And then we followed them for 8 to 12 months, 9 to 13 months actually, after the nerve transfer. And this is the flow chart of what we did. Now, this is the original 6 animals that we decentralized by just doing everything below L7. And they showed consistent defecation and urination postures, and so we euthanized them. There was 8 animals that we decentralized using L6 and L7. They all had problems. We had to euthanize all of them too. So we're reporting on these 19 animals that we decentralized with this compromised decentralization procedure with just doing the L7 dorsal root transection. And one of these animals actually succumbed 4 days later due to something that I did wrong with the actual surgery. But the remaining 18, 6 of them showed consistent urination postures, and we had to euthanize them about a year. So they weren't useful for reinnervation because we didn't completely decentralize them. The remaining animals, 6 showed only 1 or none during post-decentralization. So we reinnervated 4 of them, and then 2 of them we used for the long-term follow-up. And in another 6, the behaviors were coincident with bacteriuria. When we cleared the bacteriuria with appropriate antibiotic treatment based on the sensitivity, they stopped showing these behaviors. So we reinnervated 4 of them and we had the long-term controls in 2. So we had a total of 4 long-term controls and 8 reinnervated. And this is the urination postures in the animals that had to be euthanized due to consistent urination postures. This is the long-term decentralized animal. This is that one that showed these intermediate postures, and this one had a couple instances of these intermediate between defecation and urination postures. And you see this animal showed some urination postures after the decentralization, but they disappeared over the course. So every once in a while we saw one. And these are the animals that we reinnervated. And some of these, I don't have the antibiotic, but every time that we had an increased number of urination posture, we treated with appropriate antibiotics, and those behaviors disappeared. And then after we did the nerve transfer, these behaviors started to reappear. And in this animal in particular, we did filling aerodynamics on a monthly basis where we filled the bladder in the awake state. We put the catheters in and fill the bladder, and then we see whether the bladder, whether the animal can empty its own bladder. And I'm going to show you a video of that if I have time. So this is very positive. This is very, this pretty much confirms that we can reinnervate the bladder using an obturated nerve transfer. And this is behavior. So this is a sort of a global measurement of reinnervation. And we think that these behaviors is more indicative of the sensory reinnervation, although it could be a little bit dependent on motor reinnervation. But I think this is primarily a function of the sensory reinnervation of the bladder.
And Mike, the time delay from the nerve transfer to the exhibiting of those behaviors, we're talking about 3 months-ish, is that right?
You mean this period here?
Yeah.
It's variable from animal to animal, but in some animals it happens relatively quickly. Yeah, it's 2 or 3 months.
Okay.
And if you calculate how long it takes a nerve to grow back, that's about right.
Yeah.
1 millimeter per day.
Yeah, it's right there on the bladder wall. So it's like our, for the nerve surgeons in the audience out there, it's like our Oberlin procedure. It doesn't take very long for those axons to get to their target.
Yeah, well, 3 months seems like a long time for me if I wanted to make my bladder work. And so when we actually stimulated the bladder by electrical stimulation at euthanasia of the actual transferred obturator nerve, so we stimulated proximal to the nerve anastomosis, so the stimulation has to go through the nerve transfer site to reach the bladder. This is what happens if you do it to a normal animal. You get a good bladder contraction, about 20 centimeters of water pressure. For the decentralized animals, in the 4 of them, 2 of them we could not demonstrate bladder contraction, and 2 of them we got a low bladder contraction by stimulating this anterior vesicle branch of the pelvic nerve. Unfortunately, in these 8 animals, we really didn't get a lot of contraction of the bladder by stimulating this. In 2 of them, we got no contraction, and in 3 of them, we got about the same as we got with the decentralized, and in 2 of them, we got a little bit more. And what we did was we did a whole series of histological studies of the bladder of these animals, all animals in this group. And these are correlations between the nerve stimulation maximum detrusor pressure by stimulating this transferred nerve or the actual anterior vesicle branch itself in the controls. As a percent of collagen in the muscle layer, and so that's inversely correlated. So the more collagen, the more fibrosis, the less you can stimulate the bladder. But the major driver was this inflammation score, which is a sort of a semi-quantitative measure of inflammation. The greater the inflammation, the lower the ability to stimulate. And then these are mostly, these groups up here were these animals in this group here. And we also stained with a nerve-specific stain, protein gene product 9.5, and that's positively correlated with the ability to generate detrusor pressure by stimulating that nerve, and which is what you expect. The number of ganglion in the bladder wall is directly proportional to how well you can stimulate those ganglion and mediate the bladder contraction. So then we stimulated the spinal cord up and down the spinal cord, either the roots or the actual spinal segment itself, and we separated, we did every level, but that's an awful lot of data to show. So we separated that into just L7 to S3, which showing the maximum detrusor pressure produced by stimulating any of these spinal segments, or L1 to L6. And as you can see in the decentralized or the 8 decentralized and long-term decentralized and then reinnervated with obturator nerve transfer, L7 to S3 couldn't produce any bladder contraction. Whereas in a normal animal, that's the major innervation of the bladder and produces about 30 centimeters of water pressure. Now, here's the problem. With the normal control, there is this little bit of innervation by L1, 2, and 3. And these are primarily L2 mediated contraction in a normal animal. In the decentralized animals, we did see some contraction by stimulating these upper lumbar levels, as well as the nerve transfer animals. So we think maybe these 3 animals are the ones that had the best motor reinnervation, and these really didn't show much difference than the long-term decentralized controls. So I just wanted to show you some of these behaviors of this one animal where we videoed him in. We videoed him in its housing cage, and this is 1 year after it was decentralized for a year, and then this is 4 months after reinnervation. This is a normal Micturition posture for this animal, and it was really we showed that this. I didn't mean to do that. Okay, so what we're doing is every month is we're doing a filling urodynamics. Now we've based this on what Chet DeGroat's group at Pittsburgh, Chang-Fang Tai has shown with cats, and they've using this to study neuromodulation of the bladder, where if you anesthetize a cat with alpha chloros, that anesthetic allows reflexes to remain intact. And so when you fill the bladder through a vesicostomy in the bladder dome, and the actual bladder neck is ligated, and they're measuring pressure through a Foley catheter in the bladder neck, but the animal can't empty its bladder. So you fill the bladder, and it reaches a certain filling where you can stop the infusion, and the bladder shows this reflex bladder contraction. We thought that would also happen in dogs until we read this paper that was published a year after I graduated from high school, actually, 1973. Whereas if you fill a normal dog's bladder that's non-sedated, you get this kind of a filling curve, and this is exactly what you would get in a normal human being. The dog is not socially acceptable for the dog to empty its bladder, so it's able to suppress its tendency to have a reflex bladder contraction, just like humans are, whereas that doesn't happen in the alpha chlorulose anesthetized cat. So we were doing these filling urodynamics to try to generate this reflex bladder contraction to see if it returned. And it turns out it's not going to happen in a normal dog. And this is how we would do it. We put them in a sling. The dog is catheterized under propofol anesthesia. Once the propofol wears off, we do the filling urodynamics. But then I was talking to my colleague, Mike Panteri, and he said, I said, what do we do about this? How do we know whether the dog can empty its bladder? He says, well, you're doing urodynamics. What do you do at the end? Well, we empty the bladder and put him back in his cage. He says, well, why don't you leave it full and see what it does with a full bladder? I said, man, he's a genius. Why didn't I think of that? I'm an idiot. Why didn't I think of doing that? So here's what happens. This is the dog, number 15, 6 months after reinnervation. And this, remember, this has been decentralized for 12 months, and then 6 months after reinnervation. It's starting to do this circling behavior. Let me turn this sound down. And what that circling behavior actually turns out is that dogs are aligning themselves with the Earth's magnetic field. They like to urinate or defecate aligned with the north and south. I don't know why that is, but they do. This is 7 months post reinnervation. Dogs are circling. He looks like it's about ready to empty its bladder. Try to empty its bladder. No. There it is. There's a behavior, but it was not productive. And over here, over here is 9 months after reinnervation. Well, that's Ecta disturbed the dog, and she was so happy to see it. She disturbed the dog. So, and later we were doing it in private, so I had a little more privacy. This is a cadaver that Justin and Mary and I have been working on in a number of papers where we show in the human that let me see. No, I can't get my laser pointer going. So here is the pelvic plexus, and this is the anterior vesicle branch of the pelvic plexus coming up from the pelvic plexus going towards the bladder. And so this shows that in a human there are, and in most of the cadavers we looked at, there was one major branch coming off of here. In this one there's two. And this actually appeared in the paper. I took this from a recording. That's why it's playing. So I'm convinced that we can do this, and we're going to find out. You guys are going to tell us at the end of next week. So I think I'm going to end there. Maybe if there's any questions, I may have taken up too much time.
Now that was actually